species I'm going to be talking on today. So the department I work in and the team I work in, we're focused on managing threatened species. So it means we need to wait for species to be listed before they can come into the program. In New South Wales, we've got 4 species that have been listed as critically endangered because of declines from myrtle rust. So we've got Lindwebia species main range, Rhodomyrtus sidioides, the native guava, Rhodamnia rubescens, scrub turpentine, and recently in 2022 we had Rhodamnia maydeniana also be listed. So once they're listed, it creates a trigger under their legislation for a conservation strategy to be developed, and we've actually got 2 years to do that, but typically it takes only a few months. And once we've gone through that process, then those species will go into the prioritisation for active funding. So the reason I mention that is one of the downsides of working in threatened species is you're working on species that have already had a really significant decline, so significant that they can reach the critically endangered criteria. So we're working on species that are often in really poor condition, and that has a lot of implications for this sort of project where you're undertaking germplasm collections. So I'm just going to run through the 3 species that I'm going to focus on today. So the 2 Rhodamnias and the Rhodomyrtus. So Rhodamnia rubescens, if people aren't familiar with it, it's a medium to sort of large tree, I suppose, up to 25 metres. It occurs in lots of different rainforest and wet sclerophyll communities. It's really widespread. It goes from the south coast all the way up into southeast Queensland. And across that distribution, it's really common and abundant. It still is to some degree, but it's declining obviously really rapidly because of myrtle rust. Scrub turpentine too was a species that was commonly used in revegetation work because it was a really hardy pioneer species. It grew really quickly and was quite tough, but yeah, doesn't get used much anymore. Rhodomyrtus sidioides, the native guava, is similar. It's got a very wide distribution and was once widespread and common across that distribution. It's a small tree, gets up to about 12 metres tall. Again, it's listed as critically endangered now. And it was another species that was really commonly used in planting because it was fast growing. So it was a really good species for a lot of dune care groups to use in their littoral rainforest work. The smooth scrub turpentine, Rhodamnia maydeniana, it's a little bit different. So it's quite restricted in its distribution. It occurs from Ballina up into southeast Queensland, and it's fairly uncommon across that range. It's also a much smaller plant. It's a bushy shrub up to sort of 4 to 6 metres tall. So that just gives you a bit of a background on where these species occur and yeah, how common and widespread they are. So one thing that's common to all of these species is they've had really rapid declines. So rapid that they were able to meet that critically endangered criteria. Excuse me. We also know that flowering and seed production has been affected by myrtle rust, and seedling recruitment's thought to be basically non-existent. The remaining wild plants we see in the wild also likely to disappear, and that current generation that we see are likely to be the last generation of natural recruits unless, you know, we have some sort of management action to change that. And also just to set a bit of context for how we've responded in New South Wales, because of the decline and that rapid decline in these species, we know we're losing lots of individuals. We know we're losing entire populations, and that loss of genetic diversity has implications for long-term recovery. So our work is really focused on capturing germplasm, usually cuttings, but also other forms of germplasm, and doing genetic sampling so that we can understand what genetic variation was out there. So on the next few slides, I'm just going to run through some of the observations I've had from the field work I've done. I'll run through those and then I'll look at some of the monitoring methodologies we've used. So this slide here is it shows a frequency distribution for height class. So it's basically just showing you what size tree we commonly see when we go out and monitor this species now. For Rhodamnia rubescens, most of the individuals we're finding are in this 1 to 2 metre high range to maybe up to 6 metres. So very small plants compared to what we would have seen. I think more commonly before Myrtle Rust, they would have been in that sort of 8 to 12 metre range. So we do still see some larger plants. I have found a couple that are up near 20 metres tall, and some of those have been quite healthy, but it is much less common to find those larger trees now. One thing with Rhodamnia rubescens, we know that the susceptibility to myrtle rust is quite variable across the range and across the species. So we do still see plants that look healthy and have a good level of leaf colour. Unfortunately, Rhodomertis sidioides is not in a similar situation. What we find with that species now is that most of the plants are small suckers. They're under 50 centimetres tall, and yeah, they're root suckers from what would have been an adult plant in the vicinity. The largest individual I've ever seen in the wild was a 8 metre tall plant, and at the time of the first visit to that site, it was healthy and had a really full cover of leaves. We were hopeful that it maybe had some level of resistance, but unfortunately when we returned 18 months later, it was heavily infected. So all of the larger plants that we find for this species tend to be really heavily infected and in really poor health. Sometimes, you know, they might have 3 or 4 leaves on them. They're not going to last for much longer. For Rhodamnia maydeniana, it's I mentioned before that it's a smaller plant. It's a bushy shrub, 4 to 6 metres high. So we're still finding individuals that sort of cover all of the size ranges that you'd expect to see in that species. We've only just started working on this species over the last 2 years. But yeah, it still seems to be around in quite a variety of size classes. In terms of infection, though, most of the plants we're finding have some level of infection or signs of previous infection, so dieback and tip death. So I'll also just run through what we've found out about clonality for these species. So it's really important for us to know if individuals are clones, not just for the ex situ collections, but it's an issue that comes up a lot in the planning system in New South Wales when these species are found on development sites. So the genetic studies have confirmed that Rhodomertis sidioides and Rhodamnia rubescens both are clonal species. And for Rhodomertis sidioides, well, we sort of already knew that because what we're seeing are these sucker fields. One thing we didn't realise, though, was how extensive those sucker fields can be. So we've had individuals that have been collected 40, 50 metres apart, and they've come off the same rootstock, so the same individual. For Rhodamnia rubescens, what we've had is a few sites we've gone to where we might walk through an area and there are lots of dead Rhodamnias or really in poor condition, but then we'll find a small clump of say 4 to 5 plants that look healthy. When we've taken genetic samples from those, we've found out that that's a small clump of plants that are root suckering, and it's also gone on to be confirmed that they had some level of resistance, which lines up with what we saw in the field. For Rhodamnia maydeniana, the genetic study wasn't able to confirm that it's clonal, but it's very likely to be the same as Rhodamnia rubescens. We've seen it suckering in the field, so we know that it does root sucker like Rhodamnia rubescens does. Survivorship is another question that pops up a lot for these species. So for Rhodamnia rubescens, we know that there's variability in how susceptible it is. The remaining plants that are out there probably will persist for quite a long time, but the main issue is they're not getting replaced. There's no recruitment, and as those large plants or the remaining plants die, then they're just not being replaced. So Rhodomonas sidioides is a bit different. As I said, it exists just in these sucker fields. So we know that those sucker fields have persisted for at least 14 years because there were instances where the adult plant died the first year that Myrtle Rust arrived in Australia, and we're still seeing suckers on those sites. So it's really hard to know. For Rhodomnia maideniana, again, we've only just started working on this species. It doesn't seem to have resistance, but it tends to be slightly less susceptible, I would say, compared to the native guava or even some of the Rhodomnia rubescens plants. So possibly, it's getting enough growth in to persist as adult plants, but again, there's no recruitment happening. And just, following on that recruitment point. So we do see flowering and fruiting for all of these, but most of the time the seed, the fruit doesn't contain any viable seed. So seedlings are not observed in the wild at all. A couple of times when we've found small plants and we've thought they were seedlings, we've actually had a bit of a dig around and we've found out that they're root suckers. For Rhodomnia rubescens, that seed failure appears to be because of a lack of fertilisation. So Karen from the Botanic Gardens at Mount Annan has done some hand pollination trials with this species and has had a really good seed set as a result of that. So these next three slides, I'm just going to run through some of the germplasm collections and where we're at with those at the moment. It's been really challenging for some of these species to actually get these germplasm collections because the material is not in great condition. And yeah, it's a real testament, I suppose, to the work that Veronica's done to actually get these plants through and propagated at the rate she has. For Rhodomnia rubescens, we've done field assessments at 48 populations up and down the coast. Initially, because we didn't have genetic data to sort of design a sampling strategy, we broke up the distribution into a number of geographic areas, and we just tried to make sure that we had some samples from each of those areas. We did pretty well. There's one spot on the north coast we missed for this species, but we've had pretty good geographic coverage. And we've collected germplasm material from 67 of those individuals. And the ex situ collection's gone now from 4 genets originally up to 43 at last count and 148 individual plants that Veronica's managing. For this species, we're taking semi-hardwood cuttings, and while we often find material that is rust-free, I mean, it's not all rust-free, but often you do find fairly clean material. It's often really thin as well, so it was a struggle, I think, to get some of that to propagate. But we've now got collections that are supported by the genetic studies as being genetically representative of that wild diversity. Similar for native guava, except for this species, we've taken cuttings as well as root suckers because a lot of them are small suckers. It's quite easy just to dig those out and to send those down to Mount Annan. For this species, we've done assessments at 47 sites, 194 individuals. We've collected germplasm from 81 of those, and the collections now down at Mount Annan have increased to 76 genetic lineages and 161 individual plants. And I think those numbers of plants are actually been dropping significantly because of the dispersed collection work that's been happening. So Rhodomnia maideniana is a species we've really only focused on in the last sort of 18 months to 2 years. We've done assessments at 25 sites, which really do cover all of the known populations for that species. We've found 123 individuals. We've collected germplasm from 36 of those, and it's been a range of types of material, I suppose. We've collected semi-hardwood cuttings, we've collected root suckers, we've collected root cuttings, and we've actually, for this species, been able to collect a lot of fruit with seed as well. So we have found some viable seed. Again, those collections have increased quite a bit over the last couple of years, and there's 40 individual plants and 91 in propagation down at Mount Anna now. So hopefully, if all of those plants in propagation survive, I think we'll be at a point where we've got adequate collections for that species as well. So the next few slides, my last few slides, I just wanted to run through how we do the infection and damage assessments. So on the screen, I've got a bit of a cutout from a document I put together with Angus Carnegie from New South Wales DPI. And this was developed for the Lord Howe Island Board, who had a detection of myrtle rust over there in February last year. So they had a lot of their staff who weren't familiar with myrtle rust going out doing these assessments. So we put together a bit of a guide for them to undertake that work. So one thing that's really important if you're looking for signs of myrtle rust damage is to focus on the new growth. Myrtle rust will only infect young new growth. It won't infect old growth. Look under the leaves as well. It often starts on the undersurface of the leaf. And I suppose realise that different species, myrtle rust can have a very different appearance on different host species and also at different stages of infection. So the easiest stage to confidently identify is obviously when you've got an active infection. You've got these bright yellow orange pustules on the leaf. It's really easy to identify. What can be harder is those early signs of infection and old infections as well. The older infections, from what I've been told by plant pathologists like Angus, the best way to be confident that old infection is myrtle rust is to look for the little blisters on the brown lesions. And they're the points where those pustules have actually burst through the surface of the leaf. And you can see that in that photo on the screen there, which is from a exotic Aotearoa or Metrosideros that was growing on Lord Howe. So when I do those field assessments, every time I'm taking a genetic sample or a germplasm collection, or sometimes just if I come across a plant and I do an assessment of it, I always record the infection and damage score. And we looked into a couple of different ways of doing this initially. Some people just do a very simple low, moderate, high system. We ended up using a 7 sort of stage rating system, and it was based on a system that was initially developed by Angus and Jeff Pegg from Queensland DAF, who did a lot of the early work on myrtle rust when it first arrived in Australia. So that 7-stage system goes all the way from a 0-0 score, which is no infection or damage, all the way up to a number 5 score, which you've got severe sort of dieback, lots of active lesions on each leaf. So those active scores are based on more than just the size and the number of lesions. You're looking at stem infection as well, leaf distortion and tip dieback. I'm not going to go through each of those descriptions, but there's a fairly sort of clear description, I suppose, that backs up each of those. And we've developed scoring guidelines as well, which I'm happy to share with anyone who's interested, that sort of look at how that presents, at least on Rhodamnia and Rhodamnia rubescens and Rhodomertis sidioides. We also record a few other measures as well. We look at general plant health, so whether or not there's minor and major branch dieback, because sometimes you'll have a plant that has no active myrtle rust on it, but you can see that there's been some severe dieback in the past, and it's more than likely myrtle rust. So we record those features of the plants as well. We'll also record whether or not a plant has new growth on it, because if we're getting no active myrtle rust and it doesn't have new growth, that's the reason why. And also, if it does have new growth, what percentage of that new growth is infected? So you might have a really high infection rating of, say, 4, but only 20% of the new growth is infected. So it sort of adds to the picture of what that plant looked like when we were taking the original collection. And just a final slide. So all of that information in the field is entered into a app that's being developed for the project. For anyone who's got any of these dispersed collections and are monitoring those, it's the same app, EpiCollect. It's a different form that's set up specifically for this project. And it's just a really handy way of capturing a lot of data on each individual plant in the field, and then it syncs back when you're in range, and you can download it as a spreadsheet. So we track that information, and it means that when the genetics team, for instance, are doing their genetic analysis, they can actually look back and see, all right, well, that plant was recorded as being myrtle rust free in the field. And also when the germplasm collections are done, we can link it back to a rating from when it was collected, which is really important for a project like this. So that's all I really wanted to run through. If there's any questions, I'm happy to answer them. And yeah, thanks for the invite to come along today and to present.
Thanks, Craig. That was great. I'm just going to stop recording now. If I can, sorry, just give me a second.